Our first speaker is Dr. Matt Sturos. He's a food animal pathologist and diagnostician. And after him, we will have Dr. Stephanie Rosso, and then we'll have Dr. Fabio Vanucci. So we have 3 talks, assuming 30 minutes per talk. So, when it gets to 27, 28 minutes, I might just stand up so that I'll give you a heads up. But with that, with no further delay, Dr. Sturos, it's all yours.
All right, thank you, Hemant. Hopefully everyone can hear me in the back. See some familiar faces here, so thank you all for coming to enjoy our talks here today. I'm going to be talking about antimicrobial susceptibility trends. This is a pretty dense topic, and so you'll find that I'm not going to go into every single antibiotic, and I'm not going to go into every single bug, just because for the sake of time. All right, so first of all, we'll talk about antimicrobial susceptibility testing. What is it? What are the different types that you can do or have done on your samples? We'll talk a little bit about MICs, or minimum inhibitory concentrations. Touch briefly on susceptibility interpretations, which are usually either made off of those MIC values or potentially qualitative type tests for some of the testing methods we'll talk about. And then I've got some bacteria-specific values from our Minnesota VDL data between 2018 and 2023 to kind of look at some time point trends. All right, so when we talk about AST, there are different ways to do it. The ones that we're probably most common with are the phenotypic methods. So this requires that we isolate the bacteria and then we subject it to some sort of selective pressures. That could be disk diffusion testing, gradient strip testing. There's broth microdilution testing. There are some other methods out there that use MALDI-TOF and incubation with antibiotics. I'm not going to get into those because they're not in widespread use, and so we won't spend a lot of time on that. But recognize that there are also other ways to look for antimicrobial resistance. So you can do that with genotypic characterization. Typically that's done either using PCRs targeting known resistance genes, or you could potentially do whole genome sequencing and look for those same resistance genes. Typically this is being done on isolates, so you still need to grow the bug. And then there is a kind of still a developing field is resistome testing, and that's looking at the original materials or that fecal sample to figure out which resistance genes might be present. The applicability of this to what that means for you as a practitioner or me as a diagnostician, how that impacts what type of treatment decisions you need to make is still kind of up in the air, but that is out there as far as things that can be done. All right, so maybe many of you are familiar with this style here. These are usually based, the 2 on the left are disk diffusion tests, and then the one on the right here is a gradient strip test. And so this is you get a homogeneous growth of bacteria on the plate, and then you introduce antibacterial impregnated either discs or strips that have gradients, different concentrations as you go along. So these right here are interpreted based on the clearing zones around the antimicrobial disc, impregnated disc. This right here can actually be interpreted to an MIC value. You find where the bacterial growth intersects with those gradients, and then you actually have an MIC potentially that you can use for that. So this is some of the benefits of this is you're using solid media. So some of the fastidious bugs, so like Glasserella parasuis, tends not to like growing in broth. So if you were to try to do antimicrobial susceptibility on Glasserella, we find that our isolation rate, or those isolates that will grow in the broth that we use, that I'll talk about in a second, is 50% or less. And so if you're going to start changing the broth, you have to be careful about what type of interpretations you're going to make, because you're potentially skewing the controls in the system. And then the system that we use in our diagnostic lab, and a lot of high throughput diagnostic labs are going to use the broth microdilution test. There are automated systems, so what we use is a Thermo Fisher sensitizer. I think that's fairly widespread in the industry. And what that is you have a 96 well plate, and then you have different concentrations of antibiotic. In this graphic, it's showing going from right to left. However, in like our commercial plates, for swine, we use the bovine porcine 6 plate that Thermo Fisher has. There's a bovine porcine 7 plate, and that one has more bovine specific, so we haven't instituted that for our pig samples. But what that means is you have the concentrations kind of run in various directions, but the computer knows where that's at. And so then you start with a control, making sure that the bug actually grows in the broth, and then titrate that out and figure out at what point do you have no more bacterial growth, and that would be your inhibitory concentration. That would be that lowest dilution at which you can stop that microbial growth. This MIC can't be determined from your disc diffusion test, so when you're putting the Kirby-Bauer discs down, you have to base that specifically on the size of the clear zone around those discs, and you can't necessarily get an MIC. If you are using MICs, you have to come up with clinical or potentially epidemiological breakpoints to be able to interpret what does this MIC value mean as far as is the bug likely going to be suppressed in the host, or is it not going to be clinically effective. So these interpretations are established by some laboratory standards organizations. So here in the United States, that is the Clinical and Laboratory Standards Institute, or CLSI. There's a European equivalent in Europe as well. And then if you're in the human side, FDA also does institute some of their specific breakpoints that they have for bugs in human medicine. Realize that these interpretations are based on the host or the species at which you get that bacteria out of. It's based on the bug, so which particular bacteria you're growing, the organ that you got it out of, or potentially the specimen type, and then the combination of all of that is what is encompassed within an interpretation. So if you're missing some of those features, sometimes they get interpreted from other species. So some of the stuff that we use in veterinary medicine has been interpreted from human side. But otherwise, what you will get is, for ours, you will typically, get an interpretation, or it'll say we don't have an interpretation from Sensititer for that. When you get those back, the typical descriptions are susceptible, so the presumption there would be that this antibiotic, when used in that animal, would cause suppression of growth of that bacteria. There's resistant, and that would be based on knowledge we would think that this antibiotic would not have clinical efficacy in that animal. And then there's I. We still could refer to that as intermediate. There are different descriptions for that. The European system uses susceptible increased dose. FDA has susceptible dose dependent. So the thought there is that potentially if you increase the amount of antibiotic you're giving, you might get an inhibitory effect in that animal, but you may not necessarily at lower doses. This type of information isn't really out there for veterinary specimens, so we don't have that information. So it just gets lumped as intermediate when you look at our information. So as I said, this data is all coming from our archives using Sensititer. Most of them is, like I said, was with the bovine porcine 6 plate. We do have some in there that are bovine porcine 7, but that won't affect the break points or the cut points on the dilutions for the bugs and drugs that we're going to look at here. So I've selected 4 different bacteria to take a look at. Streptococcus suis, because that's a fairly common septicemic bug and or respiratory bug. Pasteurella maltosida, mainly for respiratory. And then a couple of GI bugs that most of our farmers are having to deal with, and producers as well. All right, so we'll move in here. The way that I'll go through this is going to be fairly standard. We'll talk about the bug. I'll go through the different specimens that we're typically isolating this bug from. So for Strep suis, the most common source of or place of isolation is here in the lung. And then we also have liver. There's a fair smattering, if you look off to the right here, of serosal surfaces. I apologize, the letters are a little bit smaller than I was anticipating, so I'll have to describe them to you. And then we've got some others like the brain and joint. So you can see the distribution of where we're growing it from. And then of the isolates that had MICs provided, typically we're selecting one Strep suis from one pig that we're going to work up if it's one pig. And if it's a case with multiple pigs, we're going to pick one pig and usually one site that we're going to work up in the interpretation that that's very likely to be a herd level issues. We do if people want us to run susceptibilities on multiple isolates from the same pig or multiple isolates from that case, we can definitely do that. You'll notice that lung, we have almost 6,000 isolates, but we're only running 475 MIC values on that. And that has to do with the fact that you can find strep suis in the lung on low levels in a healthy pig just because it's a commensal and it can potentially end up down the airway. So we tend not to run susceptibility on the lung unless that's all we get. And so that's why you'll notice that that's different. Otherwise, most of the others kind of match fairly well at about a third of what we're isolating from that particular organ. We'll have a workup. All right, so we'll move into the drugs that might get used. So strep suis, gram-positive. We'll look at some of the beta-lactam antibiotics. So penicillin, kind of basic. You can sometimes find it off the shelf, although I think at some point it's going to go to prescription. We'll see that we have about 75% are listed as susceptible. And when we look at that over the time course, so these bottom graphs here are by year. So this is 2018 to 2023. It's relatively stable. You have to take into effect that these no interpretation provided morphed down and ended up in this susceptible category. Okay. When we look at the actual MIC values, the vast majority fall here on the lowest dilution on the plate at less than 0.12. And when we look at that over time, that hasn't changed. That's stayed fairly stable. And so at this point, penicillin would potentially cover 75% of the strep suis infections that you might see out in pigs. Moving up the chain as far as beta-lactams are looking at aminopenicillin. So this is ampicillin resistance. We have 97%, so much higher percent are susceptible. And when we look at that over time, that's constant. So there hasn't been any real change. When we look at the MIC value, again, matching with the susceptibility we have, the vast majority are falling in that lowest dilution on a sensititer plate. And then ceftiofur, this would be kind of a next bump up the beta-lactam ladder. We're at 95%, very similar to ampicillin. And again, that hasn't changed over time, stayed fairly stable. MICs has a little bit more distribution when you look at the different gradients, but it doesn't affect the interpretation. So we're bouncing along here at probably around 80 or so percent that are at the lowest dilution, and then some that are stratifying within the rest of the concentrations. All right, enrofloxacin, that is labeled for strep suis in respiratory disease. So I'm showing you that here. Realize that a lot of our stuff that we're looking at, strep suis, isn't in fact respiratory disease. So there's going to be a lot of no interpretations that are listed on there. You might think, ooh, 37% being susceptible, that's not good. Realize that that's taking a big chunk away. And so if we look at the MIC values over time, the MICs have stayed fairly stable with kind of between 50 and 60% at this third dilution, and the vast majority being below that at that dilution or lower. So it doesn't look like enrofloxacin resistance is changing a whole lot in strep suis. Fluorophenocal, that's another labeled drug for strep suis in respiratory cases. This is very similar to the beta-lactams. We have, greater than 90% are susceptible across time, so there isn't really any change there. The MICs for that are again very stable. So we're seeing, 80% or more are at dilution of 2 or less on the sensititer plate. All right, so recognize that some of those drugs are labeled, they specifically say respiratory disease. And so some of them have restrictions on what you can use off-label, and some aren't allowed to be used off-label, so you got to keep that in mind. And I'm not making any treatment recommendations here for you. I'm just providing some data that can help direct what you might be doing out in the field. All right, so in the respiratory system, we'll talk about Pastorella multocida. So where are we growing Pastorella from? Not a big surprise. We've got over 3,000 isolates coming out of the lung. The next closest would be liver. Often in pigs that have a lung isolate, we can grow some of that out of the liver. There are some other serosal organs here. So we have pleura, pericardium, and pericardial sac that are all sitting here. Fairly large chunk, and we do occasionally find it in some of the other tissues like spleen or brain. And then when we look at those of those isolates, which ones are getting susceptibilities performed. Vast majority of those lung isolates are the ones that are being tested for antimicrobial susceptibility, and then the rest of them were lower percentages, but depending on the organ, again, if we grow something out of the brain that shouldn't be there, that tends to be a big question mark for folks, and so half of what we grew out of the brain had an MIC performed on it. All right, so when we look at what might be labeled or out there, penicillin, we have 80% susceptible with a big 15% no interpretation. And you'll notice that starting here in about 2019, these all morphed down into the susceptible. So we're at about 94, 95%. So the penicillin seems to potentially be very efficacious against Pasteurella multocida. When we look at the MICs over time, they are stable, greater than 90% here at that lowest dilution on the sensititer plate. When we look at ampicillin, looks very similar to penicillin. We won't spend a lot of time here. So again, we've got 90 or more percent-ish that are in those first two dilutions on the sensititer plate for ampicillin. Then we look at ceftiofur, which is also labeled for Pasteurella, 98% susceptible, and that has stayed stable over time. So we've got pretty consistent values there. And the MICs are essentially rock solid at 97% with some smatterings in other different dilutions. Enrofloxacin is also labeled for Pasteurella multocida. Here we've got 80% that are listed as susceptible over time, and that seems to be fairly stable. There does seem to be a little bit of a bump here, and I don't know if this is an anomaly in 2023. We'll have to see if that changes at all. When we look at the MICs have stayed fairly stable up through 2022, and then there seems to be a little bit of a climb in the dilutions here for enrofloxacin in Pasteurellas in the last year. So whether or not that continues, we'll have to take a look and see. Florfenicol, that's also labeled 97% susceptible for florfenicol. Again, here in 2023, there's an odd jump. So we're bouncing at about 2 to 3% for resistant prior to that. And then in 2023, we've got almost 10% of those Pasteurella isolates that are coming up as resistant for florfenicol. And when we look at the MIC over time, we see that almost 90% typically is sitting in the first two dilutions, and that seems fairly stable here. But then in 2023, we've got this cluster that's almost 10% are at 8 or greater than 8 for MIC, so that's something that we'll want to keep an eye on and see if it continues, whether or not there might be something that we'd have to take a look at. Chlorotetracycline is also labeled, and so this has high susceptibility here, 97%. That's stable across these 6 years that we're looking at, so no real changes there. You'll notice that there is a bit of a gradient as far as the minimum inhibitory concentration for these bugs, but they do tend to have the bulk of them at that 2 or less when we look at the MIC concentrations. We'll move now to some gut bugs. We've got E. coli. So for E. coli, I've split it out for non-hemolytic versus beta hemolytic. There are some differences that you'll see in the resistance patterns between those two kind of general categories of E. colis. So where we're isolating non-hemolytic E. coli, most of it's coming from intestine, feces. We do have some systemic sites, liver, lung, there's a few brain here. And you'll notice that there's stomach contents and placenta here, so E. coli can be associated with abortions, and so there's a fair number of them that are popping up. Again, this is a 6-year period, and we've got roughly 160 or 170 fetal samples that would be represented there. When we look at beta hemolytic, where we're getting most of those isolates are what you would expect. We've got most of them coming out of intestine and feces. We've got some coming out of feedback, so those folks that are working on their herd health management and checking their feedback samples to see what they might also be introducing. We did find some of that in those samples, and we're still finding some in systemic sites, but proportionally not as much. When we look at the samples that had MICs performed on them, they follow pretty much across the board. We're testing the samples that or the samples or isolates from the organs that we're getting the most isolates out of. And that's the same for non-hemolytic versus beta hemolytic E. coli. All right, so when we look at labeled drugs, gentamicin is labeled for colibacillosis. And so we've got about, when we look at aggregate, 80% are susceptible in the non-hemolytic. And when we look in the beta hemolytic, we only have about 50%. So we're at about a 50-50 in beta hemolytics versus 70-30 in non-hemolytic as far as the susceptibility to gentamicin. When we look at that over time, non-hemolytic stays very stable, so we're bouncing at that 75-ish. There's some variation you'll see over the years. But when we look at the beta hemolytic, there's a very different pattern that is emerging here. And so what we're seeing is you start here in 2018 where we have about 50 susceptible and 50 as either resistant or intermediate. And that follows very almost linearly over time to 2023 where we've got about 30% in the susceptible category and about 70% in the resistance. So that says we've got a very worrisome pattern as far as what is happening with the prevalence of gentamicin resistance in our beta hemolytic E. coli's. When we look at the MIC values for non-hemolytic, that's very stable, but mirroring what we saw on that kind of broad categorization, susceptible, resistant, we can see that that split goes, we're losing these in this lowest dilution, and they seem to be making their way up here into the highest dilution. So that's something that is just kind of mirroring what the interpretation on the other side is. The data that's providing that is also changing. All right, enrofloxacin is labeled for E. coli use. We have a lot of no interpretation provided, and so this has to do with our sensititer information that we're being provided. And we have that on both non-hemolytic and beta hemolytic. 2018 was the last year that that was reported, and we're not going to try to interpret any of that. We do have MIC data for enrofloxacin, and for non-hemolytic E. coli's, there is a decrease potentially, but it seems to be bouncing. This is 54, 53, 51, 61, 54, 60. So that seems to be fairly stable, and these at the highest dilution we've got 30, 32, 27, 26, 30. So that seems to be fairly stable. But when we look at our beta hemolytic E. coli MICs for enrofloxacin, what you see is we've got 50% here at the lowest dilution in 2018, and then we've got probably 30 or 40% here in these higher dilutions. By the time we get to 2023, we're down to 30% at the lowest dilution and 50, a little over 50% at these two highest dilutions. So again, we see a kind of a trend towards higher MIC values for enrofloxacin in beta hemolytic E. coli that we're not necessarily seeing that same trend in. Again, we do have those isolates that are resistant or have higher MICs in the non-hemolytic, but it doesn't seem to be changing. So there may not be as much pressure on those non-hemolytic E. coli's as might be being placed on our beta hem E. coli's. Another drug that's labeled for E. coli's is neomycin. Again, you'll notice that there's a lot of suppression as far as the interpretations that's coming out of sensititer for this bug, drug, and host combination, and that's the same across the board. Non-hemolytic E. coli's are fairly similar. We've got greater than 70% almost years are in this lowest dilution, with 30% in those upper tiers. But beta hemolytic, you're noticing a trend, I'm thinking. We've got 56% to 35, and now at 2023, we've got 40% to 50% here. So there is a trend towards resistance, or you can't interpret it as resistance, but the highest MIC dilution on the plate for neomycin as well in beta hemolytic E. coli's. So there are some drugs that are labeled for certain bugs, but you have to be careful about how you interpret that. So chlortetracycline is labeled for E. coli, and it specifically says E. coli that are susceptible to chlortetracycline. So I'm providing this data here to give you an idea of how susceptible is E. coli to chlortetracycline at this point. And so in non-hemolytic E. coli, we're seeing 80%-ish are going to be resistant, okay? And when we look at beta hemolytic, again, it's going to be 70 to 80% that are bouncing here in this resistant category. And when we look at the MICs, they're reflecting kind of those interpretations. So if anything, the interesting thing is beta hemolytic, the proportion that is at the highest dilution or greater than 8 went from 85 to 70 in 2023. So maybe there's less selective pressure on those E. coli's for that. I don't know. But when we look at that, you know, that would be maybe the only good news coming out of beta hemolytic E. coli's and antibiotics. And then we're going to talk just briefly about salmonella. There's the issue with salmonella is there's not a lot that's labeled out there. Most of the labels say specifically salmonella, cholera, suis, and respiratory disease. And you'll find that most of the bugs that we're growing isn't salmonella, cholera, suis, and it's not necessarily associated with respiratory disease. So where are we growing salmonella? We're growing it out of the feces and intestine, which isn't a big shocker. Growing it out of colon, fecal contents. This one here that says enteric, that's an enteric pool. This is an enriched culture. And then the tissue pool over here would also be an enriched culture, selective enriched, so we'll do a broth enrichment. The others on here are very likely to be direct plates onto, say, brilliant green agar. And so those would be things that we're growing directly from these particular organs. When we look at which samples we're running MICs, that follows very close to what we have up here as far as proportion. It's about a third of the salmonellas that we're isolating are going to have an MIC performed on it. And so here in the U.S., salmonellas from mammals are part of an NVSL or USDA surveillance, and so we get serotype information, typically from one isolate per serogroup per case. And so when we look at the different serotypes, I apologize, this is very difficult to read. This one right here is Salmonella 4 square brackets 5 comma 12 comma or colon I colon negative. So if you're wondering what that is a single phase variant of this one over here, which is called Salmonella typhimurium. So if you put those two together, that's by far the largest group of salmonellas that we're isolating is Salmonella typhimurium and its single phase variant. Okay, we do have Salmonella cholera suis right here, but this is much smaller in proportion to everything else we're growing. And then I only did N of 50. There were like 30 different serotypes that I couldn't fit into the image, so I just suppressed it to isolates that had greater than 50 for those serotypes. And then when you look at, of the serotypes, which ones are we running MICs on? And there's like 10 from each serotype that didn't get susceptibility, but for the most part, if we're going to run a susceptibility, we're going to also run serotyping. All right, so those drugs that are labeled, chlortetracycline is labeled. For those that are susceptible, you'll notice here that we're at about 65, 70% are going to be resistant. The MICs reflect that. Again, it does seem like there's a shift, so we've got 8% at this kind of 8 to 10% here, and then you come down to 2023 and we're almost at 22 or 24% that are at lower dilution, so maybe there's a little bit different pressures on these gram negatives for chlortetracycline. Fluorphenicol is labeled for cholera suis. When we look at that interpretation, we've got greater than 50%, I take that back, greater than 80% are going to be in the resistant or intermediate categories, and that hasn't changed dramatically over time. When we look at the MICs, again, you'll see this kind of cluster up. That's where those intermediates are coming in and makes it a little bit harder to interpret, and that hasn't changed over time. So gentamicin is not labeled for salmonella, but you might potentially be using gentamicin for a GI problem that you think is E. coli. Might turn out to be something else, so I included that here. And when we look at the resistance patterns, we started off with about 70 to 30 as far as susceptible and resistant, and then we've progressed to almost all resistant for gentamicin. When we look at the MIC values, you'll notice that there's a dramatic change in concentrations through almost all coming back at greater than 16 micrograms per mil. Neomycin, again, this is not labeled, but you might be using it for E. coli. And this, again, most of them are going to be classified as resistant with MICs to back that up. Again, there's a change as far as the proportion of lowest dilution versus highest dilution over time. So I do need to acknowledge the VDL clients that submitted their samples. so that we could grow these bugs and then also perform the susceptibility testing so that you could have an idea of what's going on, but that also does provide us some data that we can look at over time and hopefully pass back to you. The bacteriology staff are the heavy lifters here, right? They're the ones that are doing the plating, selecting isolates, running MICs, and so they're really the workers that made all of this possible. So we don't have that much time, maybe 1 or 2 questions. If you need to, you can contact me. My email is right here, or you can search for me on the VDL website. Hopefully the others won't be as dense for numbers. All right, if there's no questions, I think we can move on. Dr. Rosso will be up next.